Assalamu alaikum. I'm Professor Dr. Haidar Jawad Mubarak. This is a presentation to the topic of comparative anatomy of the nervous system and part of the curriculum to the second semester of the first class Medical College Al-Nahrain University. As an introductory subject to comparative anatomy of the nervous system, we must say that the basic structure of the nervous system in vertebrates is formed of two types of cells, nerve cells and supporting cells. The nerve cells are called neurons and the supporting cells are called neuroglia. The neuroglia are not only providing support to the nervous system but also provide nutrition and provides insulators in between the neurons. The basic structure of the nerve cell, which is the neuron, is shown in this figure. As you can see that the basic structure of the nerve cell is formed of a nerve cell body from which many short processes that are originating from the body. These short processes are called the dendrites, and there is only one long process originating from the body, which is called the axon. The axon is surrounded by Schwann cells that form the myelin sheath on the axon. And you know that the electrical impulses or the nerve impulses passes from the dendrite to the body and then to the axon, to the terminal part of the axon. The neurons, which is the nerve cell body, have mainly two functions. Neurons either conduct nerve impulses or electrical impulses, as we said, from the dendrites to the body and from the body to the axon and then from the axon to the termination of the axon. Or the nerve cell have another function in addition to conduction, is response to stimuli. For example, we may have here stimulus of touch. The stimulus of touch will be transmitted to the dendrite, to the body, and then to the axon. So the nerve impulse are either conductive or for the nerve impulses or responding to the stimuli. One type of the nerve cells that are responding to stimuli are nerve cells in the retina. We have neurons in the retina of the eye, in which when the retina is exposed to light, the nerve cells in the retina will respond to light, forming electrical impulse of vision. According to the number of processes, the nerve cells could be classified into three types, according to the number of processes originating from the nerve cells. These three types are the following. If you have only one process originating from the body, this is unipolar neuron or nerve cell. This one process later will divide into axons and dendrite. While if the nerve cell have one process which is axon from one side and one process which is dendrite on the other side, this is bipolar neuron because it has two processes originating from the body. The unipolar have one process that divides into axon and dendrite. The bipolar have one axon and one dendrite, so it is bipolar. If this nerve cell shows many dendrites but one axon, this is multipolar neuron. Later on we will hear in anatomy and in other topics the term nerve, ulnar nerve, radial nerve, various nerves in the body. What is the meaning of nerve? The term nerve refers to a bundle of axons that are connected together by a connective tissue. So when we find a number of axons that are surrounded by fibrous tissue, this is a nerve. And the nerve may be related to transmitting sensory information to the brain, or it convey or transmit motor impulses or electrical impulses of motor activity from the brain to the muscles, or the nerve may contain some of the axons which are carrying the sensory impulses, and the other axon carry motor, and some of them carry sensory and some of them carry motor. So it is called mixed nerve. So later on you will find that some of the nerve, which are collections of axons surrounded by connective tissue, are called motor nerves, while other nerves are sensory nerves. And sometime you will have a nerve which have some of the axons are sensory and the other are motor, and it is called mixed nerve. According to the physiological function, nerve cells or neurons could be classified into 4 types. If the nerve cell is related to transmitting impulses to or from a viscous, for example, stomach, heart, lung, this is a viscera, so it is called visceral nerve or neuron. But if a nerve cell or neuron is related to transmission of impulses to a skeletal structure or from skeletal structure, this is called somatic neurons. So we have visceral neurons and somatic neurons. In addition to that, the term afferent refers for a neuron that is functioning for receiving sensory impulses, and efferents are neurons that are related to motor function. Therefore, somatic afferent means that this type of a neuron is related to transmission of sensation from a somatic part of the body, just like the skin or muscles, bone, joints, and so on. And actually, one of the sensations that is transmitted from the muscles or joints or bones is called the proprioceptive sensation, which is meaning sense of position. When you are sitting or standing, you don't need to look at yourself to know that you are standing or sitting because you have proprioceptive sensation in the muscles, joint, and bones. So somatic afferent neurons are related to sensation from the body. Somatic afferent and efferent neurons, not afferent, efferent neurons, are related to motor activity to the skeletal muscle. So these neurons are related to motor impulses that make the muscles contract. Visceral afferents are related to transmission of sensation from the viscous, for example, from the heart, from the liver, gland, and so on. While visceral efferents are nerve cells that are related to activity of motion or motor from a viscous, for example, smooth muscles of the viscera, of the gland, of the heart, and so on. So these are the 4 types of neurons according to the physiological function, while these are the 3 types of neuron according to the number of processes originating from the cell body. Generally, the vertebrate nervous system could be described to be formed of 2 parts, the central nervous system and the peripheral nervous system. Central nervous system is a term referring to the brain and the spinal cord, while peripheral nervous system refers to the terms to the cranial nerves and the spinal nerves. And of course, I think that many of you know that the cranial nerves originates from the brain, while the spinal nerves originates from the spinal cord. And now we will discuss each of these parts of the nervous system. Starting with the brain. The brain is defined as the anterior part of the central nervous system. Generally, the brain is described to be formed of 3 vesicles in early embryonic life. These vesicles are forebrain, midbrain, and hindbrain. These 3 brain vesicles are seen in the brain of the embryo, of early embryo, and also are found in the brain of the early vertebrates. And these three brain vesicles were later on divided. That the forebrain will divide into two, which are called telencephalon and diencephalon. The midbrain will remain as itself, and the hindbrain will divide into two vesicles, which are metencephalon and myelencephalon. Therefore, the brain will form two vesicles, the forebrain, telencephalon and diencephalon. The midbrain will remain as itself. The hindbrain will form metencephalon and myelencephalon. So we will have later on 5 brain vesicles. And these 5 brain vesicles later on will give the brain, which is part of the central nervous system. So we will discuss these vesicles. First vesicle, which is a primary vesicle, is the forebrain. A synonym for the forebrain is prosencephaly or prosencephalon. This forebrain, the prosencephalon, as we said, divides into telencephalon and diencephalon. And this is the brain of shark, while this is the brain of human. And you can see that both of them have a forebrain, which is a prosencephalon, that divides into the violet or the blue telencephalon and the yellow diencephalon. The telencephalon will form the cerebrum of the brain, while the diencephalon will form many structures. This is the yellow diencephalon, will form many structures that are called thalamus, epithalamus, which means over the thalamus. Epi is over. Hypothalamus, which means below the thalamus. Subthalamus is also deep to the thalamus, and metathalamus, which means on the sides of the thalamus. So the telencephalon will form the cerebral hemisphere, the diencephalon will form thalamus, epithalamus over it, hypothalamus below it, subthalamus deep to it, and metathalamus on the sides of the thalamus. Meta means on the sides. This is a figure for the brain of a human. The pineal gland here is the epithalamus. Also, it is called epiphysis. This is it, the pineal gland. The pineal gland have a function that is related to pigmentation of the skin in lower vertebrates by controlling the activity of the melanocyte cells of the skin. While in higher vertebrates, including the human, the pineal gland, the epiphysis, which is the epithalamus, have a role in regulating the biological rhythm of the human or higher vertebrates. And what is meant by biological rhythm, that it relates the activity of the human or higher vertebrates related to the surrounding environment. This is the pineal, which is the epithalamus. The hypothalamus, which is this, which is below the thalamus, is related to the function of temperature regulation, water balance, appetite, blood pressure, sexual behavior, and sometimes, and also it is related to some emotional behaviors. The thalamus is a major relay station for sensation from all parts of the body. We don't want to pass into the function of the subthalamus and metathalamus because it is rather complicated, and you will study it later on with the neuroanatomy. So that is all about the first brain vesicle, which is the prosencephalon, the forebrain, that is formed of telencephalon, that is the cerebrum, and the diencephalon, that is the thalamus and related structures. The second brain vesicle after the forebrain, which is the prosencephalon, is the mesencephalon, which is the midbrain. This is the midbrain. The midbrain has a dorsal part which is called tectum. This is the dorsal part of the midbrain, which is called tectum in a human, and a ventral part which is called tegmentum. The third primary brain vesicle after the midbrain is the hindbrain, and the hindbrain is divided just like the forebrain. Of course, the hindbrain is called rhombencephalon, and it divides just like the forebrain into an anterior part which is called metencephalon and posterior part that is called myelencephalon. The anterior part, which is the metencephalon, will form the pons, that is the pons, and the cerebellum. This is the cerebellum. While the posterior part, which is the myelencephalon, will form the medulla oblongata. And this is the most caudal part of the brain. More caudal to the myelencephalon, which is the medulla oblongata, will be the spinal cord, which is the second part of the central nervous system. The central nervous system is formed of this structure, or all of these structures that are called the brain, plus the spinal cord is the central nervous system. You can see this figure is a summary to the brain vesicles. Initially, you have forebrain, prosencephalon, midbrain, mesencephalon, hindbrain, rhombencephalon. These are the most anterior part of the neural tube during development, and they are the most anterior part of the central nervous system in primitive vertebrates. Later on, the prosencephalon, the forebrain, will form telencephalon and diencephalon. The telencephalon will form the cerebral hemisphere, while the diencephalon will form the thalamus, epithalamus, hypothalamus, subthalamus, and metathalamus. The midbrain, which is the mesencephalon, remain just like a mesencephalon only, while the rhombencephalon, which is the hindbrain, will divide into two: metencephalon anteriorly and myelencephalon posteriorly. The metencephalon will form the pons anteriorly and cerebellum posteriorly, while the myelencephalon will form the medulla oblongata only. And therefore, the three brain vesicles will form 5 brain vesicles. More caudal to these 5 vesicles is the spinal cord. And you can see the derivatives are enumerated here. The telencephalon form the cerebral hemisphere. The diencephalon form thalamus and its related structure. The mesencephalon, the midbrain, only form midbrain mesencephalon. The metencephalon form the pons anteriorly and cerebellum posteriorly, while the myelencephalon form the medulla oblongata. And the meten and myelin are parts of the hindbrain. The telencephalon and diencephalon are parts of the forebrain. More caudally is the spinal cord. And the spinal cord plus these 5 brain vesicles is called the central nervous system. Now we must discuss the subject of the evolution of the brain. Generally, during development, the phylogenetic behavior of the brain shows tendency to increase the size of the forebrain, mainly the telencephalon. And therefore, if you compare the brain of various vertebrates which are at various stages of evolution, you will see that the difference between them is mainly in the size of the telencephalon. And you can see that the human have the maximum size of the telencephalon, which is cerebral hemisphere, which is very much developed. And this increase in the size and development of the telencephalon part of forebrain is always associated with complexity in the behavior of the vertebrates and the need for muscle, for more complicated muscle control. Therefore, during evolution, you may see that the cerebrum, which is the telencephalon, consists of 2 hemispheres. Each hemisphere is formed of 2 regions, dorsal and ventral region. The dorsal region is called the pallium, which is this one, and the ventral region is called the subpallium. The pallium is formed of a medial, dorsal, and lateral part, the subpallium, is formed of striatum and septum. And what's going on that during evolution, the cerebral hemisphere must be modified according to the behavioral complication of function and according to the complicated need for motor activity. So some of these will disappear, the other will be enlarged, the other will be fused, resulting in a much developed telencephalon and cerebrum. The subpallium, which is formed of striatum and septum, usually the septum will form the limbic system that is related to regulation of emotion and memory that is important for the complicated behavior, while the striatum will form the so-called basal ganglia that is related to control of complex motor activity or complex movement. Higher vertebrates. The spinal cord. The spinal cord is the second part of the central nervous system plus the brain. It is located cylindrical as a cylindrical structures inside the vertebral canal. Its length inside the vertebral canal varies between the vertebrates. In vertebrates with well-musculated tail, the spinal cord extends into the tail, so that you can see that there is caudal part of the spinal cord. While in vertebrates that they don't have tail or have poor tail, the spinal cord is shorter than the vertebral column. For example, in a human, the spinal cord ends at the level of first lumbar vertebra, and so there is no caudal part of the spinal cord. Spinal cord is formed of cervical segment, thoracic segment, lumbar segment, sacral and coccygeal segment only. You don't have caudal spinal cord just like in the vertebrates with well-musculated tail. If you have a cross-section in the spinal cord, you will see that the section of the spinal cord shows in the center H-shaped gray matter, which is formed by nerve cells, surrounded by white matter, which is formed by nerve fibers, which are axons, surrounded by myelin sheaths. And because the myelin sheath surrounding the axon is phospholipid, so it is called white matter because the lipid appears white. While in the center is an H-shaped gray matter because it does not contain myelin sheath and it appears gray in color, so it's called gray matter. The H-shaped gray matter is formed by right and left limbs. Each limb is formed of a ventral motor horn and a dorsal sensory horn, and these two limbs connected by gray commissure. The gray commissure contains a central canal that contains cerebrospinal fluid, CSF. The ventral motor horn of the H-shaped gray matter contain therefore neurons that are related to motor function, while the dorsal sensory horn of the H-shaped gray matter contain neurons or nerve cell bodies that are related to sensory function. The white matter, as we said, contain nerve fibers, that meaning the white matter contain axons surrounded by myelin sheath, and that's why it's called white. These axons or nerve fibers are either descending from the brain to the body, and so these are called descending motor axons or descending motor nerve fiber. Or the white matter contain axons that are ascending from the body to the brain, and they are called ascending sensory axons or ascending sensory nerve fiber. As you can see that the spinal cord give rise to roots, which are ventral root and dorsal root. The ventral root is related to the ventral horn of a gray matter, and therefore the ventral root is motor in function, just like the ventral horn. While the dorsal root is related to dorsal horn of a gray matter, therefore the dorsal root is sensory in function. and you can see that the dorsal root contains an enlargement which is called dorsal root ganglia or spinal ganglia that contains many nerve cells also. The ventral root and the dorsal root unite at the level of the intervertebral foramen to form the spinal nerve. Therefore, the spinal nerve is a mixed nerve because it is formed of a ventral motor root and a dorsal sensory root. It is a mixed motor and sensory spinal nerve. In earlier evolved vertebrates, you may not have a union between the dorsal sensory root and the ventral motor root. This is the situation in human and higher vertebrates. The ventral root, which is motor, related to ventral horn, which is motor, and the dorsal root, which is sensory, related to dorsal horn, which is sensory, and this is the dorsal root ganglia, which contain nerve cells. The sensory dorsal root and the motor dorsal root, the motor ventral root, which is in higher vertebrates and human, unite at the intervertebral foramen to form the spinal nerve. And we can see that the spinal nerve divides into ventral ramus and dorsal ramus as it will exit from the intervertebral foramen. In some earlier or lower developed vertebrates, there is no fusion or union of the dorsal root and ventral root. And the dorsal root is not only sensory, it is a mixed motor and sensory, while the ventral root is only motor. And in lamprey fish, you may not only this change is found, it is a early evolved vertebrate, the lamprey fish. In lamprey, you don't even have a dorsal root ganglia in lamprey. In others, you may have dorsal root ganglia, but the roots are not uniting, just like in human and higher vertebrates. As we said, the spinal nerve, after formed by union of the ventral and dorsal root, exit from the intervertebral foramina, and it will divide into ventral ramus and dorsal ramus. This figure demonstrates that the dorsal ramus, which is this one, and this is the ventral ramus, sorry, the dorsal ramus supply the skin and muscle on the sides of the back only, which are called axial muscle and the skin, while the ventral ramus distributed in a long distance to skin and muscles of the lateral and ventral parts of the body, including the limbs, that are called hypaxial skin and muscle. Spinal nerves number in human are the following. You have 8 cervical spinal nerves, of course 8 pairs because they are on the right and left side, 12 thoracic, 5 lumbar, 5 sacral, 1 coccygeal. So the total number of spinal nerves in a human are 31 pair. Next to the spinal nerve, we will talk about the cranial nerves. Of course, the spinal nerves are part of the peripheral nervous system, and the cranial nerves are also part of the peripheral nervous system. Spinal nerves originated from the spinal cord, while cranial nerves originated from the brain. Cranial nerves in higher vertebrates, including the human, are 12 in number. But in lower vertebrates, as in agnathian and some fish and amphibia, the cranial nerves are only 10. But in higher vertebrates that are called amniotes, as a human, for example, mammal, birds, and reptile, all of these having 12 cranial nerve, and they are called amniotes because they have amniotic cavity in the embryo during their development. The name of the cranial nerve is as following. They are 12 in number. The first cranial nerve is the olfactory nerve. It is related to olfaction, the sense of smell from the nose. The second cranial nerve is the optic nerve that is related to the retina for sensation of vision. The third cranial nerve, called oculomotor nerve, it is motor to move, responsible for movement of the eye, and therefore it is motor to muscles moving the eyeball. And the same of this movement is for the fourth cranial nerve, which is the trochlear nerve. Also this nerve supply muscles moving the eyeball. The fifth cranial nerve, which is the trigeminal nerve, is a mixed nerve. It is sensory from the head, receives sensation from the head, and motor to muscles of the jaw that moves during mastication. The sixth cranial nerve is motor nerve, also moving the eyeball, so it is motor to muscles of the eyeball. The seventh cranial nerve is the facial nerve. It supplies muscle of the face, and also it is sensory, carrying taste sensation from the mouth, from the tongue. The eighth cranial nerve is the vestibulocochlear nerve, because it is related to sensations transmitted from the inner ear, that is balance and hearing. The non-cranial nerve is glossopharyngeal nerve, because it is a mixed nerve related to carrying sensation of taste bud and also responsible for motor supply of muscles of the pharynx. The 10th cranial nerve is called the vagus nerve because it has a very long course. It is a mixed motor and sensory to the viscera, including viscera of the thorax and viscera of the abdomen. The 11th cranial nerve, which is called the accessory nerve, this cranial nerve supply muscles in the neck, which are sternocleidomastoid and the trapezius muscle. The 12th cranial nerve is called hypoglossal nerve because it lies below the tongue and supplies the muscle of the tongue only, so it is a motor nerve. In summary, the factors that are related to the description of comparative anatomy of the nervous system, which are the FEV, the function, the environment, and the presence of vestigial organ, documents that the human nervous system is very well developed in comparison to lower vertebrates, and this evolution, the high development or high evolution of the nervous system in a human, is related to the complicated pattern that the human behavior is required, and also due to the complicated pattern of motility, not only behavior. The human needs complicated behavior and complicated motility for functional adaptation. This higher evolution, in summary, for the nervous system of a human may be enumerated in 4 points. Number one, that the evolved forebrain in a human is very advanced, namely the telencephalon. The telencephalon is very well developed in a human, and that's because the complicated behavior and complicated motility needed by the human requires the development of the telencephalon, mainly the cerebrum, and that is well documented by the higher mental functions that is seen in the forebrain of the human. The second point is that the human nervous system shows a shorter spinal cord as compared to other lower vertebrates, because the spinal cords in a human is shorter than the length of the vertebral column. It ends at the level of L1 vertebra, while in lower vertebrae, especially the vertebrates with very muscular tail, the cord extends into the vertebrae of the tail or in the tail. The third factor is that the spinal nerves are mixed in a human nervous system. It's mixed motor and sensory in function. And the last point is that the full sets of 12 cranial nerve are seen in the nervous system of a human, because also to a complicated pattern of behavior and motility functional requirements that necessitate the presence of a full set of 12 cranial nerves. And as you can see, the comparative anatomy of the human in relation to other vertebrates elaborates the well development of the human, but we have to consider that earlier development of the nervous system is similar between the human and other vertebrates. If you know that many of the researches, for example drug research or even microbiological research or other research, are usually done on lower vertebrates, research on rat or on rabbit or else, and the results then will be tried on or experienced on a human. That is because in early stages of development, all vertebrates having the same stages, the same morphological pattern of early development, but later on the development of the CNS, for example, in this topic, goes according to the functional need, according to the environmental need. Some of the organs will well developed, more advanced than the others, while the other structures will will show vestiges, will have a minimal evolution. That's all about comparative anatomy. Thank you very much.